Hello, everybody, and welcome to today's webinar. My name is Karen O'Hanlon-Court, and I am the Editor-in-Chief here with CRISPR Medicine News in Denmark. We are delighted to welcome you to this webinar today about CRISPR-based control of insect-borne diseases. Our speaker for today is Omar Akbari, Professor in Cell and Developmental Biology section at the Division of Biological Sciences at the University of California in San Diego. Omar Akbari received a BS and an MS in biotechnology from the University of Nevada, Reno in May 2005. In December 2008, he received a PhD in cell and molecular biology, also from the University of Nevada, Reno, where he studied transcriptional regulation during development while working with Professor Robert Jewell. He then joined the laboratory of Professor Bruce Hay at the California Institute of Technology as a postdoctoral scholar, where he used principles of synthetic biology to develop population control technologies for animals. In 2015, he became an assistant professor of entomology in the Center for Infectious Disease Vector Research at the University of California, Riverside. In the fall of 2017, he then joined the faculty as an assistant professor in the Cell and Developmental Biology section within the Division of Biological Sciences at the University of California, San Diego. In 2018, he co-founded Agrigene, a biotechnology-based startup in San Diego. In 2019, he was promoted to associate professor with tenure and to full professor in 2021. If you have a question for Omar during his presentation, please type it into the questions box on your screen and we'll get to them after the presentation. Hi, Omar. Thanks for joining us this afternoon for me, but very early morning for you. So thanks for making the extra effort. Without further ado, I will now hand the presentation over to you, and I'm looking very much forward to hear about your work.
Hi, Karen. Yeah, thank you so much for the super kind introduction. I'm really excited to be here today with CRISPR Medicine News to present on some of the work from our lab at UCSD. I'm going to share my screen now, so give me 1 second here. Perfect. All right, you should be able to see that now. Can you see that okay?
Looks good, yeah.
Great. I'm going to turn my video off so you can now focus on the presentation, and I'll turn it back on during the questions at the end. So let me do that. Great. Okay. All right. So, well, thank you so much again for having me here today. Today I'm going to talk about the efforts that we've had to use genetic control technologies and to control wild populations of mosquitoes. And I'm going to just start off the talk today by giving you a broad overview of the various pathogens that mosquitoes transmit. And then I'll lead into some of the current control practices for mosquito control that are used today. And then I'll lead into some technologies that we are developing that use principles of synthetic biology to provide advanced genetic control of mosquitoes to impair their ability to transmit pathogens. And I'll focus the technologies we're developing on, particularly on CRISPR. So there's two main categories of systems I'll talk about. One is gene drive. I'll talk about the goals, types, and mechanisms. And I'm going to focus the presentation on one technology which we think can be used in the field in the near term, which is a confinable homing-based gene drive, and I'll explain what that is. And then I'll move on to a new type of system that we developed to provide advanced genetic control, also using CRISPR, which we call precision-guided sterile insect technique, or PGSIT for short. All right, so mosquitoes are essentially the world's deadliest animals. This is a slide that comes directly from Bill Gates' notes, and it shows you that compared to all other animals, mosquitoes kill more people on Earth than any other animal. The closest next animal is a human, which kills around 580,000 people per year, and that's mainly due to war. And the reason why it's so deadly is because it transmits so many different pathogens that can affect human health. We have pathogens like malaria, dengue fever, West Nile virus, yellow fever, Zika virus, etc. It's estimated that half of the world's population, roughly 3.2 billion people, are presently at risk of being infected by one of these deadly pathogens. Malaria alone kills about 1,000 people per day, and that equates to, most of those are children under the age of 5, and that equates to about a child dying every 2 minutes. So since this presentation started, a couple of children have already died of malaria. It's a huge problem. Dengue fever, we have more infections. We have about 390 million infections per year, with fewer deaths, around 50,000 or so deaths. On the bottom are pictured the three notorious vectors for transmitting malaria in Africa. In the middle is Anopheles gambiae. It's the prime vector of malaria in sub-Saharan Africa. And then on the left is Aedes aegypti, and on the right is Aedes albopictus. These are the two prime vectors of dengue, chikungunya, yellow fever, and Zika virus. So I thought I would put this article in here because it's very timely. We are in a pandemic, the COVID pandemic. It's affected everybody throughout the entire world. This is an article written by David Lazarus, which came out last year, June 25th, 2021. And it talks about how in a way we got really lucky with COVID, in that while it is transmissible through air droplets, it's not transmissible by mosquitoes. So we dodged a huge bullet here. What if it were also transmissible by mosquitoes? What would we do then? And I think this helps emphasize the point that we need to develop, better technologies for combating, mosquito-borne pathogens. So what are our approaches to combat mosquito-borne pathogens? And really, our first line of defense is really to reduce contact. We can do that by using insect repellents. We can sleep under bed nets. We can use lures and traps. We can modify the environment. We understand that mosquitoes prefer to lay their eggs in standing water primarily. So if we can reduce the occurrence of those places where they lay the eggs, we can reduce populations in that way. We can kill the larvae using insecticides or larvicides, or even killing the adults. We can use pathogen removal drugs, things like artemisinin, which work well for the malaria parasite. And there are new genetic technologies that have been developed, for example, Oxitec or the Wolbachia approach, which are being used in the field today. Now, while all of these approaches are really important for having a sustained control of mosquito-borne pathogens, they all have issues. On the top left, the use of repellents and bed nets, these require a continuous application, and they need to be distributed to the people that need them, and the people need to use them. On the top right, modifying the environment, is difficult to do this because oftentimes mosquitoes lay their eggs in very small bodies of water. So it's really hard to remove all potential breeding grounds. And when you have larger breeding grounds, you could result in ecological implications if you were to try to remove standing water. On the bottom left, insecticides and larvicides, these things do work, but they're temporary solutions. They're expensive, and unfortunately, mosquitoes have evolved resistance. And this is also true for pathogen removal drugs. The mosquitoes have evolved, and the pathogens have evolved resistance to these. On the bottom right, genetic technologies we use today, they're not particularly sustainable. So what we do need is to develop new technologies to combat mosquito-borne diseases. I want to point out here that mosquitoes are an obligate vector for transmission of these deadly pathogens. And what I mean by that is, if you have a wild mosquito, the wild mosquito is going to go and bite an infected individual. It's going to then incubate the parasites or the virus that it takes up from that infected individual for up to 8 to 10 days. And then it's going to transmit those to the next unlucky individual. So this is a very simple chain of transmission. And the question here is, how do we block it? And really the holy grail is to develop effective vaccines. If we can vaccinate everyone for all these different pathogens like malaria, Zika virus, et cetera, then we can potentially block this chain of transmission. An alternative approach is to engineer mosquitoes that are unable to transmit these pathogens. And the approaches that we take in our lab relate to approach number 2, and I'll talk about that. So over the last 20, 25 years, there's been some really exciting research in terms of mosquito biology and development of tools for genetics and manipulating pathogens. Now, here are a couple of my favorite examples, and this is work led by Marcela Jacobs-Arena and Tony James, in which they engineered Anopheles mosquitoes to be unable to transmit the malaria parasite. And this work was followed up in Aedes aegypti mosquitoes to block the transmission of dengue serotype 2, and this was using an RNA interference-based approach. So when our lab first started back in 2015, actually we started our lab at UC Riverside, and it was an exciting time, but also quite a scary time. It was exciting that we were starting a new lab, and CRISPR was also the technology that everyone was talking about, and it was scary in that on the news all we saw were pictures of babies like this because we were in a pandemic at the time, which was transmissible by mosquitoes, which was the Zika pandemic. And so the first actual experiment our lab took on, we opened the doors, was to try to tackle the Zika virus directly in the mosquito. And what we did was we engineered mosquitoes. So on the top right here, you can see these engineered mosquitoes that have these red fluorescent protein glowing in the eyes. We engineered the mosquitoes to express a polycistronic chain of engineered microRNAs, which were each targeted to the Zika viral genome. At the time, the Zika viral genome was sequenced, so we had that information. We knew there was about 10 protein coding genes, which 3 of which were structural, the other 7 were non-structural. And so what we did was we designed our microRNAs to target these different genes in these different locations here, and then we encoded them to be expressed in the genome of the mosquito. And we use a promoter here that expresses in response to a blood meal. So as soon as the mosquito takes a blood meal, this promoter becomes active in the midgut. It then expresses all these different microRNAs, which get processed. And if the Zika virus genome is present, then it can go and block the replication of that. And so we made these mosquitoes, and then in collaboration with Prasad Pradikar and Chunhong Chen, we did these transmission assays here, where we took wild type mosquitoes or mosquitoes that had our cassette in it. And what we found was in the saliva, which is where they would be able to transmit, we saw a complete elimination of the virus in these mosquitoes that had our cassette. So this showed that we could actually engineer mosquitoes to block transmission of Zika virus. We then followed up on this work to develop mosquitoes that could not transmit dengue virus. And at the time, there was a paper that came out by James Crow at Vanderbilt, which showed that there was a broadly neutralizing monoclonal antibody present in humans that could block the replication of all 4 serotypes of dengue virus. So what we did is we engineered this monoclonal antibody to be a single chain antibody, and we expressed it in the mosquito, again, made transgenic mosquitoes, and then we infected them with the different dengue viral serotypes 1 through 4, and then found that our mosquitoes were basically unable to transmit. So if you look at the last, which is extracted saliva, we had pretty much 0 transmission of the different dengue serotypes. So taken together, what these last several slides show is that we have this ability to completely harness the mosquito immune system and prevent the transmission of malaria, dengue, and Zika virus. All right, so if we want to prevent insect-borne diseases, the first step is to engineer mosquitoes which are pathogen resistant, which I've shown you that we've been able to do. The next step would be to take these mosquitoes with these engineered genes in them and replace the wild mosquito population with these engineered mosquitoes that cannot transmit disease. And the hypothesis here is that we should, if we can replace these mosquitoes, this should equal less transmission by mosquitoes, which should equate to less human disease. So this is kind of how I think about this. And here we have the world, we have a factory here where we can sort of mass produce our engineered mosquitoes. And the goal would be to release these engineered mosquitoes into wild disease transmitting populations. And then over time, these mosquitoes hopefully will then transmit these genes into the population and will eventually reach a point in time, hopefully within a short number of generations, by which every mosquito in the wild actually now encodes these genes and is now unable to transmit the pathogens. This is the hope. Now, the grand challenge here is, how do we force these pathogen-resistant genes into wild populations and have them take over? And really, the problem here is a problem in The problem is that these genes that block pathogen transmission are likely to result in a fitness cost to the carriers. So here we have our wild mosquitoes, and then we have our engineered mosquitoes that encode our pathogen-resistant genes, and these, just the encoding these pathogen-resistant genes, can result in a fitness cost. They're not going to be as healthy. They're not going to survive as long. They're raised in a laboratory. How are they going to compete with these wild disease-transmitting mosquitoes? And the truth is, they're probably not. So the solution is to link these genes for pathogen resistance with a gene-drive system. And gene drives have essentially this notorious ability to spread into a population, even if they decrease the fitness of the animals in which they reside. So over the last, since 2009, I have been working on developing, and our lab as well, developing a number of different types of gene drive systems that can be used to spread genes into wild populations. The first was a Medea system, which we built alongside Bruce Hay at Caltech. We followed this with a underdominance maternal effect lethal system, and then an engineered reciprocal chromosomal translocation system. And in 2020, we used CRISPR to develop an engineered reciprocally isolated, reproductively isolated species system. And then finally, we've been working on CRISPR homing-based gene drives, which is something I'm going to talk about today. So today's talk is going to talk about CRISPR homing-based gene drives, and then this non-gene drive system, which we call precision-guided SIT. All right, so CRISPR-Cas9 gene drive. Well, gene drive has something that is a concept that has been talked about for many years, over 50 years. But CRISPR really accelerated the development of gene drive systems. And homing-based systems were first described by Austin Burt back in 2002. And initially, these things were being developed using older technologies that can target DNA, like talins or zinc finger nucleases. And they were getting some promising data with those technologies. But of course, when CRISPR came out, the entire field switched over to using CRISPR, and CRISPR just made it a lot easier to target DNA. And essentially, to build a CRISPR homing-based gene drive, it's a very simple set of components that are needed. And really, in a diploid organism, which has 2 chromosomes, what one does is essentially encode the expression of Cas9 in the germline, and then also encode the expression of a guide RNA in the germline. And of course, the guide RNA is going to direct Cas9 where to cut the DNA. And then one can link a cargo to this as well. The cargo could be an anti-malarial effector gene or an anti-dengue effector gene, like I showed you in the previous slides. So if you link them together, then they'll basically be inherited together. So once you encode these in the genome of the animal, the cells, the animal cells now encode the editing machinery. So in the germline, when these get expressed, they actually then can cut the opposite chromosome or the opposite allele, make a double strand break, which then will have to be repaired via either homologous recombination or non-homologous end joining. And if the repair pathway is homologous recombination, then one could get copy of the gene drive to the opposite chromosome, thereby converting what was a heterozygote into a homozygote, as pictured here. And this process can continue on generation after generation after generation. And what ends up happening is you get sort of rapid spread into a population of the gene drive. Now, gene drives have been talked, there's two different types of gene drives that have been discussed very widely. One type is population modification kind of gene drive. And what this system can do is basically from a very small released population, these can rapidly spread into a wild population and eventually reach a point of fixation where every single individual in the population will encode the gene drive. In principle, if the drive system works perfectly, a single organism release into a wild population could spread that trait throughout the wild population. So it's a very cost-effective way of modifying an entire population. So if you linked a engineered antipathogen gene to the gene drive, then you could potentially make the entire population disease resistant, for example. And one point here I want to make is that ecologically for population modification, the wild population would still exist, but it would be completely engineered at the point of fixation. So the other type of gene drive system that has been widely described is a population suppression kind of gene drive. And the way this system works is, again, a small release into a population will result in gradual spread into the population to the point of fixation, by which then the population will decline rapidly and eventually crash out, as pictured here with the red line. In principle, with this type of system, a single organism release could result in this outcome. And ecologically, this is much different than population modification in that once you reach fixation, the population then declines and suppresses or eradicates. And so what you end up with is a complete void of that animal from the population. And there's a couple different ways to design these. Really, the way the population crashes after fixation is that these gene drives are designed to drive into a recessive gene that's important for female fertility or viability. So at the point of fixation, all the females are either dead or infertile, thereby the population declines really quickly and crashes out. Now, these two broad categories of gene drive, population modification, population suppression, are quite exciting. However, they do have issues. Now, the fact that they can spread so quickly from a single organism release into a wild population creates logistical issues. Firstly, it makes the system very invasive in that it can spread beyond borders. It makes it somewhat uncontrollable after you release it. It's very difficult to control. It makes it non-reversible. So once you do release it and modify the population, the only way really to convert the population back would be to release another system that could then act as a countermeasure. And we've published on systems which we call erasers or e-chasers in collaboration with Ethan Beer's lab that could be used for that purpose. Because of these potential issues, it makes it very difficult to measure risks. And the open question right now is are these approaches really regulatable? What are the unintended consequences? Would the public accept this type of approach? And we talk about many of these issues in this paper here. On the bottom, if you're interested, you can take a look at this paper. So because of these issues with gene drives that can spread rapidly, what our lab has been developing is trying to sort of tamper the ability of a gene drive to spread and to make them somewhat self-limiting. And we were really trying to provide all these desired features which would give sort of more control of the system, make the system somewhat confinable, make it reversible, make it safe, but yet still effective. And so what we thought of was this idea of basically generating gene drives, which we call split drives. So we essentially remove the Cas9 nuclease from the gene drive system. So we end up with two components. We have the guide RNA and the effector as one component, and then the second component would be the Cas9 and the nuclease. And this is much different than the drive on the left, which is a linked drive, which has the Cas9, guide RNA, and effector all linked together. So essentially, if we unlink these components, we could basically generate a system which would result in the self-limiting behavior, which would require multiple releases for the drive to spread and fix into a population. So we sought out in Aedes aegypti, which is the major dengue transmitter around the world. We working in this species, we decided to build a split drive system, again with the Cas9 split from the guide RNA and effector. And we designed this to target a gene which is important for eye pigmentation, to make it easy to measure phenotypes, and we used a fluorescent protein to mark the presence of the drive element. And so we could measure whether we're getting inheritance of the drive or just cleavage of a target using these separate phenotypes. And without going into all the details of this, we essentially generated our system. And what we found is that when we cross these together, the drive and the Cas9, we could get homing efficiencies around 84% and cleavage efficiencies upward of 100%. And this was stable across multiple generations. And so, taking the data that we generated from this, we collaborated with John Marshall from UC Berkeley to do this mathematical modeling to determine how our system would behave if it were released in a wild population. And what we found is that, if we take our split drive and we perform 10 releases of homozygous males, we're only releasing males because males don't bite or transmit pathogens, and we release these at a 1-to-1 total population frequency, we found is our drive system, so pictured in pink here, rapidly spreads to near fixation. So upward of 97% of the population has at least one copy of our drive, and that persists for up to 3 years above 85%. And the system is not predicted to spread into a neighboring population, assuming a 1% migration rate. Now, if we compare these to if we just release the refractory gene alone, so if we release our anti-dengue cassette alone with no gene drive, again, 10 releases of homozygous males at a 1-to-1 population frequency, what happens is the refractory gene goes up in frequency because we're releasing quite a large number of them, and then rapidly kind of falls out and again doesn't invade a neighboring population. Now, if you compare that to a linked homing drive, where the Cas9 is linked with the guide RNA and the effector, and in this case, you just do a single release, what happens is this rapidly spreads into the population and then starts to decline, but then spreads into neighboring population upwards of 67% and continues spreading. So this kind of illustrates that we've developed a split drive system can be quite effective. It's going to require more effort, more releases, but that will give you more control and less sort of spreading into neighboring populations. So now the system is ready to be linked to an effector. I'm going to kind of pause here for a second and kind of outline what we've talked about so far. So far we've talked about confinable population modification. I've shown you that we've been able to engineer Zika and dengue resistant mosquitoes. We've developed highly functional germline expressing Cas9 lines, which we use to develop our split gene drives in Aedes aegypti with homing efficiencies upward of 84%. And we've demonstrated drive stability over multiple generations, and mathematical modeling shows that this system can be quite effective. So more pending experiments on this system are sort of long-term population experiments. linking an effector to the drive. And then we also are working toward generating more evolutionarily stable gene drives. I didn't go into this, but in this drive system that we developed in Aedes aegypti, we did find resistant alleles, even in our small populations in the lab, which we think will be problematic in terms of spreading into a wild, more diverse population. So we want to try to find ways to sort of mitigate the problem of the generation of resistant alleles for drive spread. And so we recently came up with a new system, which we call Home and Rescue, or HOMER. And here are two papers here, one from our lab and one in collaboration with Ethan Beer's lab, in which we developed these HOMER drives, which are population modification gene drives, which are quite evolutionarily stable and could be the next generation of gene drives for mosquitoes. So I'm going to switch focus here from gene drive to a technology which we are really excited about in our lab, which also uses CRISPR. And this system actually relies on a very old technology that has been used for over 50 years to control insects in the wild. And that is sterile insect technique, which is a method of biological insect control. And really the way this system works is quite simple. You take your insect of choice and you mass rear it in a facility. You then separate the males from the females and you sterilize the males. Traditionally, it's done using radiation. So they irradiate these adult males. Then they deploy them as adults into a wild population. These males that are released into the wild population then find females and they mate with them. And because of the fact that they're sterile, they don't produce any progeny. And over time, after repeated releases, the population can decline and suppress and even be eradicated. And there's been quite a number of success stories of using this technique for control. On the bottom of our few different examples of the screwworm, the Mexican fruit fly, and the medfly. There are some limitations to this approach, and really the main limitation is that irradiation reduces the fitness of the released males. When you irradiate them, you're randomly inducing chromosomal aberrations like deletions, point mutations, inversions, translocations, which can compromise the fitness of these males and make them less effective at finding mates. Unfortunately, this approach, traditionally has not worked well for mosquitoes. There's a lot of papers that back in the 70s and 80s where they worked extensively to try to use this approach for mosquitoes and failed. There are new efforts to try to use this and with better results, so time will tell. But again, they're using this old approach of using radiation, which does compromise the fitness. Finally, you must release adults for the system, and which makes it difficult to scale. Now, here's just one example of SIT being quite effective over a very vast range of land to control the screwworm. And this happened started in the 1950s, where sterilized male screwworms were released throughout the United States. And then eventually it became clear that these were coming back in to the United States from Mexico and Central America. So what then happened is these sterilized screwworms were released in Mexico and then different parts of Central America, all the way down to Panama. And at Panama, they created basically a permanent barrier where they essentially released, up to 15 million sterilized male screwworms per week, even today, to prevent the screwworms from coming back up from South America back into Central America and back into the United States. So essentially, we've seen, using the SIT approach for the screwworm, we've had complete eradication of the screwworm from all of this vast range here. And here's an article about this in the Atlantic, which came out in 2020. And in this, they talk about how even today there's planes that drop 14.7 million sterilized screwworms over the rainforest that divides Panama from Colombia. And again, that's to prevent them from coming back up from South America back into Central America. All right, so we knew about this technology, and we also have been working quite a lot with CRISPR. So we wanted to see if we can merge these two technologies together. And one of the additional problems with existing SIT technologies is that it oftentimes requires the production of a factory which can produce these sterilized males as adults, which then have to be manually released. And this creates a problem in that, for many insects, you can't really transport adult males very far because they're fragile. And when you're trying to do this in mass, it becomes logistically challenging. So what ends up happening is we have to build factories kind of all over the place to create and deploy these sterilized males. So what we want to try to do is try to optimize the logistics of this by potentially developing a technology that could require the development of one very large factory that then can produce these sterilized males, but instead of producing them as adults, produce them as eggs. And then since eggs are very robust and durable, one could then potentially transport those eggs using drones to different places around the world to lead to population suppression. So again, using CRISPR, we came up with this technology called PGSIT, and here are two papers which we published. Initially, we published this in flies, and then just last year, we published it in mosquitoes, where we suppressed mosquito populations using this approach. And the approach is extremely simple, and it relies on using two components, one of which is a guide RNA line. So we have a line that expresses guide RNAs, and these guide RNAs are designed to target different genes. And one type of gene is a gene that's important for male fertility. The other type of gene is a gene important for female viability. And essentially, when you make this guide RNA line, you encode multiple guide RNAs targeting those two types of genes, and then you could homozygose that. And it really doesn't do anything to the fitness of the animal because the guide RNAs actually can't operate unless they have Cas9. The other line is basically just a Cas9 expressing line. And what you do is you make this Cas9 expressing line that expresses Cas9 both in the germline and in the soma. And you homozygose it. And again, without the guide RNA, it doesn't do anything. So you have these two different strains which you mass produce that are both homozygous. And then what one does is essentially cross them together. So take the females from one, the males from another in the factory, cross them together. And essentially all the eggs that are developed from that cross will only hatch out as a sterilized male, because all the females will die during development, all the males will be sterilized, and one could hatch out sterile males. So what this does is it creates an opportunity where you could actually deploy eggs into the environment and ensure that you get only sterilized males. And these eggs can be deployed via joins. And this makes the process of using SIT logistically very scalable, because every single female you sort for mosquitoes, each female lays upward of 300 eggs, right? So you have you're going to get 300 times the progeny as you would normally when you sort a single male for an SIT type application. A single male results in a single male release. A single female sorting results in 300 progeny, 150 of which are going to be sterilized males. It's environmentally friendly because it doesn't use insecticides or larvicides or anything like this. It's evolutionarily stable because we've actually separated the components from the system. We can homozygose them, raise them in mass, and the only time they come together is when you cross them, but then the outcome of that is a dead end, it's a sterile male. It can be quite effective and it's controllable. So in Aedes aegypti, which again is the world's most prominent dengue vector, we've been able to use this approach to sterilize 100% of males, convert females into intersex, and also convert females into flightless females. And so here's on the bottom, we have a picture here of a wild type male, a wild type female. and then we have our intersex. And in the intersex female, we have longer maxillary palps, and we have malformed genitalia, which also make them sterile. So I have a little video here which will show you the production of sterilized males and flightless females. So I'm going to push play. So what you're looking at here is an outcome of a cross between a Cas9 expressing line and a guiding expressing line. This guiding expressing line is designed to target a gene important for female flight. And so all the males hatch out and they can fly, they're sterile. All the females hatch out and they get stuck on the water like this because they cannot fly. They can move their wings around, but they can't fly because we're targeting a gene that's important for female flight. It's a myosin gene, important for the muscle of for their flight muscle. So essentially, this is our first attempt at using PGSIT was to make females flightless, but again, we've moved on from this to convert them into sterile males. You can see them trying to crawl around and move their wings, but they can't because the gene has been disrupted using CRISPR. Okay, that video is kind of fun. The song is fun. Again, it was our first attempt at using PGSIT and Aedes aegypti, targeting this gene important for female flight. And what we found is all the females could not fly. So if we wanted to use that approach for mosquito control, what we could do is actually have a production facility where we mass rear our two separate strains, our guiding line, Cas9 line, both homozygous. We could use automated sex sorting machinery, and there's several companies that have developed these to separate the females from the Cas9 line and the males from the guiding line, cross them together in a different cage, produce eggs. Again, each female is going to produce up to 300 eggs, 150 of which are going to be sterilized males. We could release these eggs into the environment using drones, or we can release them into pods, kind of like what Oxitec is doing. You can drop the eggs into here, and only the males will be able to fly out from this little hole here. And on the bottom here, this is just showing that, of course, this is going to require multiple releases. But as you continually release these sterilized males into the environment, the population will decline, and you could even reach a point of eradication. Now, this technology, it's really great because it uses CRISPR. And the cool thing about CRISPR is CRISPR has been basically shown to work in every animal and species it's been tested in, to my knowledge. And in mosquitoes, it's also been tested in almost all of these different species pictured here and shown to work. So the fundamental mechanism of PGSIT has already been shown to work in all these species. Now, the only thing one would have to engineer is, pick the target genes. So a gene important for female viability, a gene important for male fertility. Fortunately, the genes that we've chosen are conserved. And so we anticipate those will work quite well. And then again, the last thing is to genetically encode everything, but transgenesis works quite well in mosquitoes. So we don't envision that to be a problem. So this platform technology could be transferred to different mosquito species quite quickly. And we actually estimate that, it could take us up to, 18 months from, just picking our species to developing a functional PGSIT system that could be used to control and suppress the population of that species. Quite exciting. So this is kind of my summary slide here, kind of running out of time here, but going forward, I showed you that we can develop, cassettes that can block transmission of dengue virus, that can block transmission of Zika virus. We want to kind of move forward with this, and we're really excited about using CRISPR RNA targeting systems in mosquitoes. We've tried these already in flies, for example, using CASRX. And what we found was we had really high rates of collateral off-targeting. So when we actually have our target present, we could actually, what we found was we can kill the flies, even if we're targeting something like GFP, but only when the target is present, and that has to do with collateral off-targeting. So what we think is if we can leverage that by designing a CRISPR RNA target system to target a virus, then potentially when the virus is absent, the system would be inactive, but when the virus is present, then we would get cleavage and potentially collateral off-targeting, which could lead to complete death. And this could act as like a Trojan horse. So this is something our lab is working on developing. We're also continuing our development of confinable gene drives in mosquitoes. We're working to link effectors. We're looking to develop more effective systems that could spread and persist, but in a confinable way. So with the hope of transitioning these to the field at some point. And then of course, we're working on precision guided SIT in mosquitoes, but also in crop pests. We think this could be a very valuable technology that could be regulatable to control and eliminate populations. So with that said, I just want to thank everybody from my lab listed here, our graduate students, our lab techs, our undergraduates, all of our collaborators, and all of our funders that have funded the development of the different projects and the work that has gone on in our lab. And also want to thank you for your time, for coming to this webinar. And I will be happy to take any questions at this point.
Hi again, Omar, thank you for a really interesting presentation. That's very fascinating stuff. I've been collecting questions in from the audience, and I have a few questions myself. So I'll just start with asking you a question about the CRISPR gene drives. It's not a topic that I know a whole lot about, and I'm just curious to know how many, or if even, are there any CRISPR gene drives out there now? How far is that in general?
Oh, that's a great question. Yeah, so there's several groups that are developing CRISPR-based gene drives, and in different species. I think the furthest along is probably going to be the work that's done in mosquitoes at this point. And there are groups that do want to transition their technologies to the field, but again, the regulation is a bit unclear, and there are differing opinions on this. So if you talk to different people, they'll have their own opinion. Everyone has their own opinion on it. And, some think that, one would have to, if you're using a gene drive that can spread beyond borders, then one would have to get permission and approvals to use such a drive and basically anywhere it could potentially spread into, right? And so I think that's kind of, what people are discussing right now. It's unclear. Nobody has taken a gene drive to the field. Mm-hmm. Yet, and so it's really unclear whether it's something that would be regulatable, would be accepted by the public. So I think a lot of it is just lab. It's all been done in the lab. There's a lot of discussion on it, but nothing has really advanced further than that.
Okay, yeah, you touched on the regulation, the regulatory aspect, and that is something that we come across a lot. Most of what we cover is related to editing the human genome to correct diseases and such. And there, obviously, off targets is a massive consideration in terms of getting regulation for some of these gene editing therapies. Is off targets, or how much of a concern are off targets in some of the approaches that you're using here? So both the gene drives, but also the sterile insect techniques.
Yeah, that's a really great question. So I think the off targets that you're talking about are really, when you have a human therapy, for example, then you might get off targets of, if you're targeting a specific location or gene, then you might get targeting of something else, right? Another gene or somewhere else in the genome. And that is of concern for human therapeutics. But for gene drive and for PGSIT, off targets inside of the mosquito, for example, are not much of a concern, because really those are just going to compromise the fitness of the mosquito. It might make the system less effective, but ultimately we don't expect that to be problematic. Now, off targets in terms of will the gene drive actually spread from one species to another? those are true concerns that people have. And before any type of system would ever be deployed, of course, one would have to do a risk assessment. And in the risk assessment, this would be something that would be tested, right? If can the gene drive spread to other species, and can that be tested in the laboratory? And what's the likelihood of that happening, and so forth. So those are things that would have to be tested before deployment. But I think in terms of like off targets inside the mosquito, that's less of an issue, and really something that it shouldn't be a problem for like PGSIT, for example.
Yeah, great. So obviously malaria and going after mosquitoes is one of the most obvious targets here. Where do you see that this will be most useful in certain parts of the world, or do you see that we will eventually, see these kind of approaches also outside the regions where malaria is a huge problem? And here I'm thinking specifically about other kinds of diseases that are caused by other kinds of insects. Do you think we will see this more widespread?
I think so. I think we will see more genetic control technologies being used, and many of the new technologies are using CRISPR. With gene drive, again, yes, the prime system that's being pushed forward is for malaria, but our lab is developing gene drives for dengue virus, for example. So if we can develop something which is safe, effective, controllable, and there is a need, which we know there's a need, and we can find a location which is interested in doing an experimental trial, I think we could actually push these forward to the field and potentially reduce transmission that way. So I think we are going to see more and more of this. I'm not sure exactly how fast it will be because of the gray area in terms of regulation and social acceptance of these types of a technology. But I think technologies like PGSIT, for example, are something that we could actually really get a regulatory approval and social acceptance for really quickly, because it's really just a release of a sterile male that doesn't persist. So it's very safe and still effective. So I really think with CRISPR, the first types of technologies I believe which we'll see in the field first are going to be confinable, self-limiting type technologies. Yeah.
So obviously the PGSIT technique is really neat. And when I first read about it, when we interviewed you last year, I thought this is so clever. I mean, it's hard to see the drawbacks of it, but are there actually any drawbacks to this approach?
I mean, I think the only potential drawback is going to be the cost, right? It's going to cost a bit more money and effort to do repeated deployments of a technology. But I think we think the cost of this will be cheaper than the cost of insecticides and other things that are being used now, right? So which also cost money and also have cost to the environment. And so this is a very environmentally friendly way of controlling a specific population, species specific. And we also think the costs are going to be cheaper, but it's going to require a little bit of effort, right? You're going to have to do repeated deployments. It's not like a gene drive where you can just say, hey, I'm just going to do a single release and my problem is solved, which is kind of the fantasy of what people think. It's going to be a silver bullet. The problem's going to be over. I don't necessarily believe that. I think in the lab we do see the gene drive is, first of all, it's not perfect, and it breaks easily. We see resistance, right? We don't know what the effectors that I've shown you, like for example the anti-malarial, anti-dengue, anti-Zika. Yes, they work great in the lab, right? But how are they going to work in the field? And one of my big fears is you're going to have evolution is happening, right? You have, malaria has been around for millions, billions of years, right? And so it's evolving. And, and so when you release this and put a selective pressure, right, to prevent the transmission from the mosquito, you're now forcing a bottleneck for the parasite it has to evolve in order for it to survive. Yeah. And so when it evolves, what is it going to evolve to?
Yeah.
And how do we know it's not going to be something worse, right? And so that's one of my biggest fears. And
Wow,
I think
yeah.
that's something that needs to be tested before
Yeah.
before trialing such a technology.
Wow, that's really interesting aspect. And it's difficult to predict what would happen, actually. It's like looking into a
Yes.
crystal ball. I guess really it's a numbers game, isn't it? It's about, can you release enough of these eggs fast enough to actually get this level of suppression that you need to eradicate? Do you think that this has the power to eradicate malaria? Or will it be in combination with the other approaches?
I think what we're thinking right now is that, like, in urban settings, one could do releases of PGSIT, for example, and protect like the urban environment, and then potentially maybe suppress, eradicate from the urban area. But then as you do that, you kind of spread further out and then further out, right? You'd have
Yeah.
to keep moving your intervention.
Mhm.
And one of the things that we think is important is having what we call a sensing loop. So where one uses a trapping technology that could enable sort of real-time data analysis on the population, what's going on with the population of the mosquitoes, right? So if you have these traps all throughout your target site, then, and those traps are feeding data into a cloud in real time, telling you, okay, well, here's the current population density in
Mm,
this location, and so you take that and you have a map, then you can use that to tailor your deployments. And so
yeah,
you have more effective deployments. You're deploying where the mosquitoes are, you find
yeah,
out where they're coming from, and then you deploy back. And so we think a combination of a sensing loop using very sophisticated trapping systems and software alongside deployments, we think could result in elimination from urban settings relatively in a relatively cost-effective way and safe and species-specific way. So we think that definitely is true, and we're we won't know until we try it, but that's kind of what we think.
yeah. No, that makes a lot of sense. Thanks. We have a question in here from Brett. First of all, great presentation and good music choice for the video. I also thought that was great. So could the technology be used to stop diseases for wildlife? So he says that mosquito-borne disease has killed numerous bird populations in Hawaii, New Zealand, and even on the Galapagos over the last century. For example, the honey creeper birds in Hawaii. He says they're island populations that might have potential for effective testing also. And since the mosquitoes are not native to Hawaii, they could be followed in this remote area and likely would have little ecological effect since they're not native. Do you have any comments to that?
That's a really, really great question, and something we're really interested in doing. And so right now we actually have Culex-Q, which is the main vector of avian malaria, and in our lab. And so we're working on developing PGSIT for Culex-Q. And we think that once we have that system up and working, we agree that these island settings are perfect trial sites for such a technology. And if it's effective enough, we could result in eradication of Culex-Q, which would prevent avian malaria transmission to those bird populations, and potentially, hopefully, save those birds before they become extinct. That's something we completely agree with, and we're working on.
Yeah, thank you. Another question from the same person. So using the merged CRISPR sterile insect technique system with homologous genes may cause mass sterilization in the worst case scenario. Are there preventative measures possible to ensure the system doesn't cross? Perhaps the CRISPR system could be made in such a way that it's only active during a certain stage of development with proper placement of the insert.
So I think what he's trying to say is, if you got a translocation, right, and created an event where the Cas9 and the guide RNA became linked, then potentially you can get this. I think it's really unlikely because we've unlinked them, right? So we have Cas9, different chromosome than the guide RNAs. They're both homozygous, and when you cross them, the only result, the only thing you get is a sterile male, right? So you don't have a chance for that sterile male to pass on any of these genes to the next generation. So it's a really robust and stable way ensuring the system always works, because you don't give it an opportunity to break. It only has that one chance. And even if you get sort of, let's say you do get a non-sterile male, that non-sterile male is going to be hatched among, however big the release size is, millions of sterile males, right? So the chance in which it's going to be able to pass on are incredibly low.
Yeah.
And so we're not too worried about that, but we have been thinking about developing suppressible PGSIT systems. And so we have developed a new system, which we call temperature inducible PGSIT, where we do actually keep the Cas9 and the gidronase in a single line, again, on different chromosomes. And in that system, we can homozygose it and have it inactive at low temperature. And then essentially, when we want to generate sterilized males, we just raise the temperature, heat shock it, and then we get sterile males. And that actually works really well. We've only developed that so far in flies. We're working to transfer it to other species. It just takes time, but we think that may be easier to scale, because you won't have to do a cross in the factory. Instead, you just maintain one line and you use temperature to induce the activity of the system. And we found it to be quite robust. And one of the reasons why it works so well is that Cas9 alone is temperature sensitive. So Cas9 works really well at 37. It works less well at 18. So if you maintain your insect line at 18 and macerate at that temperature, the Cas9 alone is relatively inactive. And also the promoter driving Cas9 is also relatively inactive. So you have sort of 2 points where you're kind of and so we're pretty excited about that.
Yeah. Cool, thank you. The last question, we're on to the last question now, and then I'll let you go, because I know it's really early for you. Must be time for more coffee soon. So what is the next plan on the precision guided sterile insect technique project? So what is the next move?
Yeah, I mean, so we've been working on transferring the system to those 5 species I showed.
Yeah.
And right now we actually have a working system in Anopheles gambiae, where we can kill all the females and sterilize the males. And so we're really excited about exploring opportunities in Africa to do a trial of that technology to see if we can suppress populations and hopefully save lives. Our lab is really, we've worked on gene drive probably longer than most labs have, and developed probably the most diverse array of types of gene drives available today. And we're excited about them, but we do see this really obvious rate limiting step of taking the technology from our lab to the field. And because of that, we've kind of reverted back to other technologies like PGSIT, which can be quite effective and regulatable to enable us to get into the environment quicker to actually save lives, because we want to use our technologies. We want to save lives, and we see the problem in Africa and elsewhere in the world, and we want to get out there and start helping. And so we're really excited about the opportunities that exist, like, for example, the opportunity in Hawaii to get rid of the Culex Q, which is killing off the honey creepers, or people dying in Africa from malaria, or people suffering in other islands from dengue virus. So we want to get out there. We want to use our technologies. We don't want to just develop things that are cool that sit in our lab. That's not our goal. So that's kind of what we hope for the future.
Yeah, that's really interesting, and we're looking forward to seeing what happens next. And hopefully when you start to save lives, you'll come back and give us an update. We'd love to hear about that.
I would love to come back and give you an update.
Yeah, well, that's it. A lot of people saying thanks very much for a lovely presentation. I'm going to let you go now and say thanks very much once again to Professor Omar Akbari for taking the time to tell us about this fascinating work. And we thank you, the audience, for listening in and answering questions. And we hope to see you at a future CRISPR Medicine News webinar. The on-demand version of this webinar will shortly be available online on our website, CRISPRmedicinenews.com. And there you can go in and see this webinar and all our previous webinars free of charge. That's it. So thank you very much once again, and have a nice day.
Thank you so much. Bye-bye.
Bye. Bye-bye.